All right, so in this video, we're wrapping up this chapter on eukaryotic gene expression. So remind you guys, previous video was on prokaryotic gene expression. This one's going to be about how do eukaryotic cells control their protein synthesis. And so one of the things we got to mention is a lot of reasons why people should have some understanding about this is nearly every genetic condition has some error in protein synthesis, whether that be excessive protein synthesis or underrepresentation of proteins. So in terms of this, we got to at least establish a recap of what does the eukaryotic chromosome look like. So remember, chromosomes is when the DNA is really, really condensed. But chromosomes versus chromatin, they're the same thing. It's just when DNA is really condensed, it's not going to be able to be expressed. Chromatin is when your DNA gets a little more loose. So in terms of that, we now have to make a bigger issue about these little histone proteins right here. These things in yellow are histone proteins that DNA is wrapped around. And nucleosomes are going to be grouped histones together. So I wouldn't fixate so much on those, but definitely know histones. Because then that is going to play into heterochromatin versus euchromatin. So just to kind of reiterate, if your DNA is really, really condensed like this, that's what we're going to be calling it chromosomes, but when it's a lot looser, we're going to call it chromatin. So heterochromatin is when your DNA is tightly packed, you're not transcribing, which means no mRNA is going to be produced, versus euchromatin is when your DNA is a lot looser packed. Okay, that means transcription is occurring. You actually can read it. So key player in this is going to be histones and how this works. So this kind of leads into one of the greatest, one of my biggest pieces of information that I hope you guys get out of understanding protein synthesis is what's the difference between cells. And that has to do with what's called gene expression in eukaryotes. So cell differentiation sounds complicated, but it's not. It's just basically what proteins get expressed. So what I like to think of what protein is expressed is going to equal the cell type. And I should clear that up as it's the genes that get expressed, right? So in terms of this, if we all started out as a single cell, did mitosis, what makes this cell a neuron and what makes this cell a skin cell is the genes that get read. So notice that this is trying to emphasize that these two cells have the same DNA, but if I read this region of DNA, I'm going to make the proteins that make this cell become a neuron. If I read this portion of DNA, I'm going to make the cell become a skin cell. So the genes that are expressed is going to determine the type of cell. So it's all about the proteins made, okay? So this video is going to be about, well, how does a cell know actually how to turn on those proteins and how does it know when to turn off the expression of those proteins? So because all the cells contain the same DNA, it's just what DNA is read. That's going to give it a cell type. So this one is getting into what is considered chromatin regulation. So this says the structure of histone spacing matters. So these little green spheres represent the histones. So I don't want to go crazy in depth here, but at least kind of remind you of a couple of things that if we have acetyl groups here, acetyl groups basically have these big negative charges. And all you have to know is that if this is normal, right? If you added some acetyl groups down here, you started to space out your DNA, this means that it's turned on, gene expression is turned on. So that basically means RNA polymerase can actually bind and read that gene, okay? So that means you're actually reading it. If your DNA is loosely packed, we're gonna call that euchromatin, okay? So DNA is loosely packed, you are actually able to read it. Well, what makes it unpacked is adding acetyl groups, okay? Now on the contrary, what if you added in a bunch of methyl groups? Well, the more methyl groups you have, and if you forgot what the methyl groups are, they're these little CH3 methyl group is what a methyl group is. And they're all nonpolar. And basically what that does is it's going to want to compress all of the DNA together. And that lowers transcription rates. So that means your DNA is really, really condensed. So basically you just have to know is that if there's a methyl group there, you can't read it. And if it's acetylated, it's actually going to be read. So if you go back here, if you didn't catch, it had the word meth here. That just means that all of this DNA is methylated except for this one area. That's the part of the DNA that actually gets expressed to make these proteins. A different cell, that's part of the DNA is now methylated. and you're reading this part to make those proteins. So that's the difference between histone acetylation and DNA methylation. If you got acetyl groups, it spaces it out. You can actually do protein synthesis because you're reading it, versus if it's methylated, you're not actually able to express it. So this gets into some interesting things about epigenetic inheritance. The point of this is environmental factors can methylate DNA that isn't supposed to be. So things like stress, different environmental factors can actually cause DNA to be methylated that normally shouldn't be. So it's not controlled by base sequencing. That basically means the DNA doesn't change, but for some reason you're not making proteins that are needed. So think of a smoker, somebody that smokes cigarettes. A lot of their DNA gets methylated and they're not actually making a lot of collagen that should be on their skin. So that is something that is important to understand. Now, this next part is going to be on how does a cell turn on protein synthesis. Not only is it just about having it acetylated, but how does it know how to turn it on? So I'm going to jump to this little visual from Mastering Bio, and let me just pull this up right here. And a lot of this does essentially seem fake, but I assure you it's not. There's so much going on here. So let's unpack this. So right here we got RNA polymerase. In one of the previous videos, I mentioned these transcription factors. Okay. Now there's going to be a couple of new key players here. So this is RNA polymerase. We're going to have a TATA box over here in your promoter region, right, with your TATA box. These are transcription factors. They're already existing. So, right, we're inside the nucleus. So basically, we have to have these signals called activators. Activators bind to a region of DNA called an enhancer. And what it does is it bends over the DNA. These transcription factors now combine. And now basically it's just one other factor to turn on the expression of this gene, okay? So what's really new? The only new part about this was those little activators. So activators, as it says here, bind to a region called an enhancer, and then it's going to help bend the DNA to actually express it. So when you think of it, it's basically how is regulation of transcription occurring in these eukaryotic cells? Well, you have these little signals called activators. Activators bind to a region of DNA called an enhancer. It helps the DNA bend. It recruits these other transcription factors, and then this gene can get expressed, okay? Now, let me kind of show you this picture really quick. It looks a little messy, but we were doing it in class, so forgive me on that. But basically, this is kind of putting the missing piece together on a cell signaling problem. So remember cell signaling, there's 3 steps: reception, transduction, and typically you have a response. Your response 99% of the time is to make a protein, so let's see what's going on here. Your signal binds to a receptor protein. Remember, in the this is a transduction phase. This protein activates that one, activates that one, activates that one. But this last protein turns on one of those transcription factors, okay? So basically what this is saying is that, and this is great from your book, this cell, the DNA in both cells contain the albumin gene and the crystalline gene. Don't worry about what albumin crystalline is, but you just have to know that the genes are there. Now, this is talking about the lens, which is a part of the eye, and then a liver. So this is showing you that that crystalline gene is not expressed because it received the activator to read this gene, okay? Now, vice versa, if we looked at the lens, it still had the gene to make that albumin protein, but it received the activator to express this gene. So what I drew over here is that this signal can activate that gene because it activates that transcription factor. This gene gets expressed because this signal activated that transcription factor. So basically it just provides that missing link that these enhancers are there, and it's the activators that need to get turned on. So basically that's what we're talking about. The signal, transduction, the response, we're putting this together. This signal activates that transcription factor to activate that protein. So hopefully that's making some sense to you, that when you think about regulation, it's all about the activator, okay? Now, it all kind of stems to those cell signaling questions as well, okay? Now, another thing that happens when we get into eukaryotic gene expression is alternative RNA splicing. So I believe I have a video here, yeah. So cells can adjust to changes by what happens after transcription. So let's remind what happens in RNA processing. And again, this is eukaryotes only. So remember, phosphate on the 5' end, poly A tail. I snurped it, I spliced it. This is my exon, right? Intron stay in, okay? Now, what this was showing you is that from the same gene, I can actually alter my RNA sequencing. So before I show you the rest of it, what you need to understand is that the same gene can actually make different proteins. So if this was the gene, I had 1, 2, 3, 4. I have these 4 exons. If I put these exons together, exons 1, 2, and 3 on my final processed RNA, I'm going to make protein A. If I take the exons 1, 2, and 4 and I put those together, I can make this protein. So different regions of the mRNA can be spliced out. So it just depends on what the cell is needed. So this kind of shows you a couple of other things too, that the mRNA provides additional opportunities for post-transcriptional control. So just to remind you of a couple of things here, that the lifespan can change. So the longer the poly A tail, the longer it's going to be expressed, okay? So some RNA molecules can be translated multiple times. It just depends on certain signals as well as these enzymes that start to degrade that poly A tail, okay? So it just shows you that sometimes RNA can last longer than others. So this is showing you that same RNA sequence is being read again and again. So it's being translated multiple times. So an eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made, all right? So the more that gene or that mRNA sequence is read, the more of that protein, okay? Now, this one is kind of interesting. So translation of mRNA may also be blocked by proteins that bind. So check this one out. You have a protein that binds and blocks that ribosome from being read. And that's important to note out because what those are going to be referred to as these little microRNAs. So if you look here, this little microRNAs is basically stops translation. So it prevents that RNA from being read anymore. So this is called post-translational. So basically, you've made your RNA, you've made the protein, but now you need to stop that ribosome from reading it. And this is showing you get a protein that blocks it. So you get proteins that can actually block translation here. So it's really important because sometimes it takes some time to break down that mRNA sequence. This just shows you you can get things to block the ribosome from reading it. And that's those microRNAs. Now, the last little bit is talking about proteasomes because you also have enzymes that need to actually be broken down, or sometimes proteins need to be broken down and they get tagged with this molecule called ubiquitin. When a protein is tagged with ubiquitin, it signals it to actually be degraded. So basically what's happening here in terms of this, what you need to think of is if I wanted to get rid of this protein, the cell marks it with ubiquitin, and then it's going to be broken down or digested, okay? So this is showing the proteasome, and basically what happens is it ingests it, breaks it down into the individual amino acids, and those amino acids will get recycled for another protein. So that's your protein degradation. Now the last little bit of this, we'll be talking about cancer and in terms of errors that can happen. So if you think about these oncogenes, okay? So most of the time we're considered proto-oncogenes, genes that basically can stop the cell cycle if it becomes faulty. But if that gene is mutated, we call it a cancerous gene or an oncogene, okay? So basically if you had UV rays, the DNA gets damaged, that could be considered an oncogene. Now, in terms of this, there's two types or pathways really of cancer. Now, this one is showing a mutation of the RAS. So let me kind of clarify this. This is the signal, binds to your receptor protein. This is the transduction phase, and normally you make a protein that will start the cell cycle, okay? Now, this is important because this basically is providing the checkpoints to proceed, that you're able to do it. Now, notice the difference here. If you have this mutation, you no longer need that receptor protein because you have a mutated RAS protein. So that means regardless of what's going on, you are overproducing this protein. And realistically, this protein at the end is cyclin. And now you should know that the excess of cyclin, the excess cell division you're doing. So no reception is needed. The cell's constantly making cyclin. It's constantly doing mitosis. So overproduction of it would cause tumor growth. Now, another form of cancer, this is more detail on the RAS protein. But in terms of this, we also have the P53 gene. And this one is really important for us to understand, where typically if you get some type of faulty gene, right, you have a kinase sequence here this protein activates this P53 protein. And what this protein is going to do, it's going to make another protein to stop the cell cycle. So think of it this way. If you have, let's say, a cancerous cell, this protein, this P53, recognizes damaged DNA. And what it does is then it makes another protein to stop the cell cycle. So it stops that faulty cancerous cell from dividing, right? Now, if you have UV light, and let's say your P53 is mutated, there is no more P53 gene to make the P53 protein, you won't be able to stop it. So a cell is going to be doing mitosis when it normally should. So what is the difference between these two? Well, if the RAS is mutated, you're doing excessive cell division, nothing is stopping it. The P53, if this is the pathway that is mutated, the difference is that this P53 normally would stop the cell cycle and it would detect damage. But if that P53 is missing, you are no longer able to stop the cell from dividing, and it's replicating more than it needs, as well as it's replicating faulty DNA. So that would be the biggest issue, right? So that is a short explanation of the eukaryotic gene expression, and we'll see you in the next video.